All right, let's start. We have a couple of things to do. So, hello everybody. This is Gültekin. I welcome you all to today's webinar. Um, I'm one of the co-chairs of the Bioinformatics Pipelines and Data Visualization Working Group. So today, Bede will present us a practical panhuman host sequence depletion for responsible data sharing. Before further ado, um, let me give you some um, housekeeping things. So Bede will present for 40 minutes approximately. Then we will have a Q&A session. Please feel free to ask questions with a Q&A section on the bottom. This webinar is also live on YouTube. So that's almost it. So let me introduce Bede to you. So the Bede Constantinides is a computational biologist interested in the practice of genomic epidemiology and bioinformatics methods that enable it. He is a current research fellow at the University of Birmingham in Nick Loman's group and a member of the Arctic Network. Bede completed his undergraduate studies at the University of Manchester and went on to a PhD in bioinformatics supervised by Professor David Robertson. As a postdoctoral researcher, he led research into the risk to patients posed by the colonization of the hospital sinks with the drug-resistant bacteria, and later into respiratory virus transmission among healthcare workers at Oxford University hospitals. His current research focuses on fast and local bioinformatic methods for targeting genomic and metagenomic surveillance. Bede is a vocal advocate for open data sharing and standardization. And also, he's, I think, a member of our working group as well. So, before handing over to you, Bede, thanks for accepting this. And we had this conversation in the PHA conference like a couple of months ago, and now it's happening. So,
Yeah, thank you.
it's
Thanks for the
it's really
intro.
good. It's really good to have you. So in this talk, Dr. Bede will outline the risk and trades-off involved in making genomic sequence data safe for analysis and sharing, focusing on the common task and removing human sequences from microbial DNA sequencing data. So he will present software called Deacon and demonstrate its practical use for bioinformatic host depletion, as well as more generally for fast DNA sequence search and partitioning. This will be followed by discussion and the Q&A. So over to you, Bede. Thank you.
Thanks. I'm just going to try and share my screen. Um, I have shared the wrong one. Okay. Can you see some slides coming up?
Not in the yes. Now
Yeah, great.
it's in the presentation mode, right?
Great. Yeah. Thank you so much for the introduction, Gultekin. Today I am going to talk about some work I started at the University of Oxford and continued at the University of Birmingham, where I am now working with Nick Lohman. And my work in Birmingham is funded by the Wellcome Trust through an award to the Arctic 2 project. And initially, this work was funded by the UK NIHR, National Institute for Health Research. And I'm really grateful to these funders and my current and former advisors for securing that funding that's allowed me to work on this. So today I'm going to talk about three things really. Why we do host depletion, the different ways we can do it, and the approach I've developed, and briefly how to use it. Hmm. We're gonna get okay. um, yeah, so I put practical in the title of this seminar because although there's some time to talk about these methods in more detail, um, if you take one thing away from this session, it's that if you're sequencing non-human DNA that might contain human DNA, you need to think carefully about how you remove that DNA bioinformatically. Secondly, ideally, you want to use a pan-genome aware approach. And finally, sort of none of these approaches are perfect, and there are trade-offs involved that we should look into. Um, and while host depletion is particularly important before you share data, actually it should probably happen even before you analyze your data. And I appreciate some of you listening will already be very familiar with this topic in general, and that I'm preaching to the converted, so to speak, to some extent. But yeah, I've put a link to the approach I've developed, a GitHub link, that we're going to sort of briefly walk through later on at the end. And for anyone who wants to try it out while we're on this call or get ready to do it afterwards, particularly if you're on a slow internet connection, you might want to start installing it, while listening. There is an index to download, which is 3 GB, which is from a server in London. So you well feel free to download it now if you want to get ahead. And note that you will need a Linux or a Mac OS machine, or else to do that through using Windows Subsystem for Linux. Um, and there are other ways to install it, which are shown on the GitHub URL. Perhaps someone could sort of share that URL in the chat, so that people can access it when I change slide. Okay, what's the time? Yeah. So let's rewind. So what do we mean by bioinformatic host depletion? We're talking here about removing sequences belonging to a host genome in which we are not interested. We want to discard sequences belonging to the host organism. When we try to DNA sequence microbes living on or in a host organism for research, diagnostics, or whatever, our DNA libraries inevitably contain some proportion of host sequences. And often that host is human, but certainly not always. We might be studying microbes of mice or of plants and so on. And bioinformatic host depletion is really a binary classification problem. And something that makes that classification problem challenging is the high dynamic range of host DNA content across different kinds of samples that we might want to sequence. So yeah, the ratio of host to microbial sequences varies a lot between sample types. For example, for human gut samples, most of your DNA sequences might be microbial, but for human skin microbiomes, human sequences could easily make up 99.9% of sequences. So this poses a challenge for doing that for classification. And so starting with a mixture of microbial and human sequences, we attempt to filter out the human sequences. We do this often out of obligation to eliminate personally identifiable information and protect donor-host privacy. But also, human sequences can actually harm some microbial genomic analysis, such as taxonomic classification, and particularly things like de novo genome assembly. So here I've sort of defined some terminology just so it's a little bit clearer what I'm talking about when I refer to a true positive. That is a host, for example, human sequence classified correctly and removed. And when I talk about a false positive, I'm talking about a non-host sequence, such as a microbial sequence, that's been wrongly classified and removed, which in this context we don't want to happen. So if you're wondering why we should care about this, you can point to sort of a great deal of evidence suggesting that host depletion is being done poorly and can cause quite practical problems. Someone called Stephen Bush did a study of bacterial reads in public databases and found that 6% of bacterial read datasets contained enough human sequences to reveal sensitive genetic information about the host human individual, such as their genetic disorders and likelihood of developing certain diseases. And this has been echoed and supported in various studies since then. There was also quite a high-profile retraction of a cancer microbiome Nature paper where it turned out that the microbial signal that the authors claimed could be used to predict cancer was in fact not a microbial signal at all, but driven by human sequences inadvertently influencing the microbial analysis. So we sort of have two categories of things that can go wrong when you do host depletion. You have loss of privacy and loss of analytical accuracy and soundness. And especially in relation to the privacy concerns, I think we should be concerned as microbiologists, bioinformaticians, and public health practitioners, because a big privacy scandal sort of could end our ability to freely generate and share sequence data as we do now. So we need to ensure we are making our data safe to share and analyze for microbial signal. So my own motivation for developing new methods for host depletion was a concerning discovery that widely used methods are broadly destructive of microbial genomes. This figure is from a blog post of mine. Each marker on the left panel represents simulated reads for one clinically relevant bacterial genome. And on the right, each marker represents simulated reads for a distinct human genome. So the crux of the left-hand panel is that some microbial taxa are badly affected by host depletion methods, losing more than up to sort of 0.2% of their reads in some cases. And you might expect that the method in blue with the lowest false positive rate might suffer in its ability to classify human host reads on the right. And it's not, indeed, it's not the best, but it's also mid-pack. It's far from the worst. And so the method in blue is called hostile, which I developed around existing approaches, Bowtie2 and Minimap2. And it provides a favorable compromise here compared to widely used methods. And so if your priority is retaining microbial reads, then it's a good choice. And unlike Kraken 2 with its 70 gigabyte standard index, which does do better in terms of removing human reads, Hostile can run probably it can run on most laptops. And if we drill down on some of those bacterial genomes from the last figure in the last slide, and we look at those that are sort of most wounded, so to speak, by the process of host depletion, we see that it includes some really clinically important genomes like Neisseria gonorrhea and C. difficile. And we're not talking about reads randomly, evenly distributed across these genomes. We're talking about gaps in coverage, systematic gaps causing missed variant calls, broken genome assemblies, and that sort of thing. And we don't want those in our analysis. And this, this is a relevant problem that affects the kind of bacteria we care about, for diagnostics, public health, genomic surveillance, and so on. So I was really surprised by these results at the time. Why, these methods have been developed by good scientists, and they benchmark well, but they're not performing well in my hands under simulated viral and bacterial genomes. So reads that I know don't contain human contamination that you might see in kind of real world data sets. Well, it becomes kind of quite obvious why this is when you dig into the literature and look at how these methods were actually developed and how they were evaluated and what the sort of objectives, what the goals were. So some of these methods were designed with very specific applications in mind. For example, for instance, like removing host reads from bacterial reads of a very particular species, a task at which they may perform very well, but that species may not share any resemblance to the human genome, which isn't the case for all bacterial genomes. And by using these tools to remove host sequences from complex, diverse metagenomes, you are essentially misusing them. That wasn't really what they were designed for. And they weren't evaluated in that context. So perhaps it's not surprising that, they may not perform so well in that context. I think more concerningly are the papers, of which there are several, which evaluate metagenomic host depletion despite only considering a handful of taxa. And I'm of the opinion that if you advertise a host depletion approach as being suitable for metagenomes in the title of that paper, you should probably evaluate more than three viral taxa and no bacterial taxa in the case I'm thinking of which I've quoted a little part here. So as a result of quite limited benchmark data sets, the sensitivity and specificity trade-offs for many tools are, in my opinion, quite problematic. So out of this frustration and this realization, I developed Hostile a few years ago now. It's easy to use and has sort of sensible default parameters. And as shown in the earlier figure, it's got good microbial retention compared to existing methods and competitive but not leading sensitivity. And this has been independently verified in quite a few studies since. And it's sort of been adopted fairly broadly across academia and public health and has sort of 50 citations now when I checked. But it does have some limitations. Human genomes are very similar and yet different enough that when using sensitive read alignment, even when using sensitive read alignment, you miss some parts um, of diverse genomes. And so ideally, it would support pangenomes to allow it to do an equally good job for all genomes we've sequenced before. But relying on alignment makes this practically difficult. And finally, being based on alignment, it is just relatively slow, particularly when dealing with host-heavy sequence data, where a lot of computation is essentially wasted aligning human sequences only for us to then throw them away. Um, and I think it's important that something as basic as host depletion, is as quick and convenient as reasonably possible. So there are broadly two classes of approach to host depletion. Alignment is the go-to way to tell if two sequences are similar, but is compute heavy. Alignment-based approaches can involve either selectively retaining sequences or selectively removing them. An example of selectively retaining them in this manner is a tool called Read It and Keep, developed by Martin Hunt at the European Bioinformatics Institute. But this kind of approach, obviously only really works if you know what you're sequencing and you know what you want to keep. So it's not well suited to agnostic metagenomics where you could sequence anything. And the inverse is to align everything to a host genome, such as a human genome, and discard all of the sequences that stick to it. And this is how approaches like Hostile and many others work. But an alternative, sort of moving to the right of this slide, is to decompose sequences into words of a fixed length k, which are called k-mers, and see how many of them match. There's kind of a large shift towards k-mer methods spanning decades now in bioinformatics, and it's increasingly how a lot of methods work, kind of in an era of just rapidly growing data and also analytical ambition. So the problem with the k-mers is that the information they store is quite redundant. But there are elegant ways of selecting k-mer subsets that perform much better than naive random sampling, and these are often called winnowing schemes. And minimizer schemes, which are on the right here, they're an example, and they have this lovely property of given two identical sequences, you always get the same minimizers for both, and minimizers are k-mers. So you always select the same k-mers from two identical sequences. And if the sequences are similar but not the same, you select most of the same k-mers. So it's a compression technique, often called a sketch, that allows you to extract representative landmarks of your genome for fast analysis of any kind. And so minimizers reduce the number of k-mers you need to store enormously with only a modest reduction in sensitivity for sequence matching problems. And these are really widely used. And this is in part how tools like Kraken 2 work. So if we naively try to use a reader liner like Minimap2 to index 100 human genomes, naively our index would become 100 times larger and unwieldy with mostly redundant sequences. And we'd need a huge amount of computer memory and alignment would be slow. We could use a pangenome graph aligner and a pangenome graph reference, but in my testing, this is just quite a bit slower. So k-mers and minimizers, on the other hand, naturally represent pangenomes very efficiently. You can add together the k-mers from 100 human genomes, and they're only marginally heavier than a single human genome. And that's because you only need to store the parts that are different. Conceptually, this is superior in that we can succinctly represent potentially all of the reference quality human genomes ever sequenced to boost our ability to classify human sequences equitably across different human ancestries. And this is the sort of basis for the method called Deacon, which lets us search and filter and dehost FastA and FastQ files accurately and quickly. When we give it a set of sequencing reads, we check each minimizer in each FastA or FastQ record for membership in a minimizer index that we've loaded into memory containing, for example, human minimizers. If the number of hits exceeds a relative or absolute threshold, or well, actually both of them, the record is either kept in search mode or discarded in depletion mode. So a query index can be a tiny single gene or a massive mammalian pangenome, and it barely affects the speed. And it's got a command line interface, which is fairly simple to use if you're familiar with those. And you can index and filter and perform set operations on really large sequence collections in order to do things like build custom indexes for new hosts. And while built for host depletion, it's really a general purpose sequence search filter that you can use for any application. So in theory, a k-mer-based approach can be really fast, and some effort has gone into making Deacon fast in practice. So it's the first approach of its kind to make use of a highly optimized library published last year by Ragnar Groot-Kerkamp and Igor Martin, who are really talented computer scientists. In practice, this makes Deacon several times faster using something called SIMD acceleration, making use of the special vector execution units that all modern processors have, be it Intel, AMD, or ARM. So otherwise, it's built from scratch for speed and minimal memory copying, rather than wrapping existing general purpose tools, which tend to be slow. So optimizing the performance has been an interesting exercise in trial and error for me, and I'm really grateful to Ragnar, who has helped me improve the performance of Deacon quite dramatically since I first released it. And in practice, it's almost always gzip decompression or SSD speed that actually caps its performance now. And this is an old version. We're going faster now, up to about 4 gigabases per second on a 16-core AMD machine. And this is significantly faster than any other index-free search approach I'm aware of. And this is a plot showing speed on the y-axis with a number of execution threads on the x-axis, showing how Deacon can use multiple processor cores. And this is an order of magnitude faster than any alignment-based approach. And actually, its performance doesn't change for host-heavy data like it would for an alignment-based approach. And the energy usage is tiny compared to existing methods. And you could process Promethean flow cells worth of data in a couple of minutes. So the main surprise for me in developing Deacon was how accurate a k-mer-based approach can be, even when dealing with relatively error-prone and challenging for k-mer-based methods, ONT reads. Being a k-mer-based method, we can effectively choose where we end up on a sensitivity specificity axis. That's what's sort of shown here, by adjusting our k-mer length and our minimizer parameters and our hit thresholds. And this shows some of the experimentation involved in selecting the default thresholds, which are possible to change, but I would argue deliver best balance of sensitivity and specificity. So some microbes share chimeras with human genomes, and some much more so than others. In addition to tuning chimer length and match threshold, with Deacon you have the ability to mask or subtract chimeras of, for example, microbial genomes that are similar to human genomes to ensure they are retained. And this is quite a good way of reducing the false positive rate further. And it also helps clean up low complexity chimeras, such as spans of the same base, improving accuracy. And this is and these kind of operations are easy to do on the command line. Yeah, for example, if you research retroviruses which have fossil remnants in the human genome, you might find that a custom mask could improve the accuracy of your host depletion. But RefSeq viruses, the database of virus genomes, has already been subtracted from Deacon's default panhuman 1 index. So it should be good, should work well out the box for both viruses and bacteria. This plot compares the host depletion sensitivity of several methods using diverse human genomes. Sensitivity was evaluated using simulated reads made from the Human Pangenome Reference Consortium genomes. Deacon and Detaxizer seem to be doing the best in terms of sensitivity, with similar performance for Illumina in the top panel. For ONT reads below, Deacon was second to Detaxizer using a very large Kraken index. But that came at the cost of specificity, which we'll see in the next slide. So rather like I did with the hostile manuscript, I simulated a really diverse catalogue of microbes in order to validate the specificity of the approach, which I think is quite unique in this sort of field. And here we're showing sensitivity, specificity, and balanced accuracy. Balanced accuracy being the sort of black diamond, all on one plot. And in the previous slide, we saw that Kraken-based methods, the Detaxizer was very sensitive, but here we can also see that its specificity was very poor in this benchmark. And we see that basically the Kraken-based methods, both of them, so no human being another, both of them struggle with no human discarding 4% of simulated bacterial reads. So really this shows that the Kraken 2-based methods don't fare so well, at least with their default parameters, which can be changed. And for short paired reads, even for short paired reads, it has lower specificity. Deacon performs generally best except in one scenario, which is for short paired reads, where hostile has greater sensitivity, which I appreciate is quite difficult to see on this plot. So there was a lovely paper recently describing a conceptually similar approach to Deacon, but with a very different implementation, which I had the pleasure of reviewing. The authors benchmarked Deacon and showed that their approach and Deacon are both considerably more accurate than Kraken-based methods, which was reassuring and kind of validates the analysis that I was just referring to. So I should say that Deacon has improved rather a lot since this benchmark and is now considerably faster, for example, than their figures suggest. But in my opinion, in terms of accuracy, both of these tools are in similar territory, and I think are good choices for panhuman depletion. So to summarize Deacon, I think it's important that host depletion can be done well, really quickly and painlessly, even if you don't have access to big computers. There was a paper last year that proposed a method that requires essentially a sort of supercomputer to do host depletion, and which I think is a bit strange. But anyway, I think Deacon provides this. So sequence databases are getting larger, sequences are getting faster. We know that the forthcoming Roche sequencer will generate data incredibly quickly compared to existing platforms. And so, more generally, we need fast, efficient, sufficiently accurate methods for whittling down big data sets into the things we're interested in. And Deacon is equally useful for searching a gene in a huge data set as it is for host depletion. Yeah, if we can do bioinformatics on a big, expensive server, that's great. But I think it's a lot better if we can do it on a cheap laptop in the field without internet access, allowing us to sort of to decentralize analysis, which is one of Arctic Network's goals. And this method is engineered in a way that it could run on a smartphone or miniature DNA sequencer or similar in the future. So I consider Deacon now broadly complete, and I'm currently working on a significantly improved manuscript with among other things, better benchmarks. Um, and I'm also interested in making Deacon as easy to use and accessible as possible, which is where I think it could use some improvements, including for those who are uncomfortable using a command line. To that end, I understand integration with the Galaxy workflow runner is in progress. So for users familiar with Galaxy, that will soon be an option, I hope. So to wrap up, most host depletion tools perform quite poorly with complex microbial metagenomes, as they were not really designed for it. As such, care needs to be taken to balance host depletion and microbial retention. Deacon is an example of a tool designed to do this, but not alone. And I'd say the best thing you can do is to experiment with your own data and try out different approaches. But most importantly, make sure you do some form of reasonable host depletion, particularly before you share data that might contain sensitive host genetic information. And this will help ensure that we maintain the trust as a community needed to uphold local and global data sharing practices. And lots of people have contributed to this work in one way or another. Um, and I'd like to thank Nick Lohman, Derek Crook, Ragnar, Igor, John Lees, Jeremy, and Hugh, as well as some third-party sort of code contributors, Peter Menzel, Louis Maddox, and Tommy Macklin. And I think now is a good time for questions. Thanks for listening.
Wow, it's amazing, Bede. Thanks for the presentation. Can you hear me?
I can, yeah.
Yeah, okay. Thank you. And thank you for this comprehensive and insightful presentation about Deacon, especially the metagenomic host depletion. You gave a lot of different comparison about the host depletion approaches. So I want to try that after this presentation. So this is really valuable for everybody around the world, I think. So before going to a Q&A session, let me give some time to the people who wants to ask questions through the Q&A part. And I want to ask a question, if I may. So where's the naming coming from? I know in bioinformatics, naming is a very important part of the tool development. So how do you name your tools?
Um, Erratically, I would say. So I guess with the first host depletion tool I developed, it had the word host in it. Uh, that was part of the reason. And this one has, I guess, is a play on decontamination. Um,
Oh, I see.
but I also like the idea of like a deacon is a kind of a humble servant. Um, so yeah, there was there was a bit of that in there as well.
Thank you.
I'm not a particularly religious man though.
Yeah. Yeah, but naming is a very important part of the tool development process, I think. Okay.
Thank you. Shall I look through? Shall I try and answer these questions in the chat?
There are a couple of things. I can read it to you out loud for everybody to read. So a lot of thank yous. Some people wants to test it with their own data. And one question, by how much do you believe this approach will make dehosting faster in comparison to, say, standard read mapping against the human genome?
So yeah, it's an interesting question. So I think it depends on quite a lot of variables. And for example, if your data is stored on a hard disk, particularly on a networked hard disk, maybe over the internet or something or over a network, then it might not make that much difference if that's the bottleneck. But if your data is on, say you have a modern laptop, say you have a MacBook or something, which has quite fast storage, then potentially 10, 20, 50 times faster. Um, yeah, it depends what the bottleneck is, but on a modern machine, it can be quite dramatic.
So it's actually it's the same about the what about memory footprint? You partially
Yeah.
answered this question as well.
Yeah, no, it's
Okay.
a great question. So the default index, which I think which isn't really compromised by memory, the memory, it's not trying to achieve a smaller memory footprint. Um, that essentially requires about 5 GB of RAM. However, I've shown that you can get extremely close to that with a much smaller index. So it's relatively straightforward to make an index that's half the size, but still has 99% of the accuracy. So yes, it's 5 GB is the memory footprint as it stands now, but that's not fixed.
All right, thank you. Let me check again. Okay, there's another question from Tracy. Is there a methodical way to go about choosing the parameters like chimer size?
Uh, Good question. It's so no, I don't think there is the answer, but someone else might disagree. Um, I think it really depends on what your priorities are with the data. I chose for this analysis, I chose an objective function of given my data set, which is simulated reads for a thousand bacterial genomes, very high quality reference grade bacterial genomes, and viral RefSeq, which is only a fraction of the sort of viral universe, but is nevertheless you know, a somewhat representative sample.
Mm-hmm.
My objective function was balancing for it was about 30 simulated human genomes, some of the most diverse human genomes we have, was balancing precision, sensitivity and specificity
Yeah.
for classifying those two data sets. So essentially a very complex metagenome, and I'm considering both Illumina and ONT, and I have chosen parameters to suit both and
Mm-hmm.
hit thresholds. It's not just Kmer, it's not just your Kmer and your minimizer parameters, it's also your hit thresholds, absolute and relative hit thresholds. I've kind of tried to achieve balanced accuracy. So in some applications, you may want to focus more on on sensitivity than specificity, and you could adjust those parameters accordingly. And I'm very happy to chat about how to do that in a somewhat disciplined way. But yeah, there's no magic answer. It depends on your data and your priorities.
Mm-hmm. Yeah, thank you. Thank you for the answer. Quick question. Is there any other indexes that we can apply for other hosts, I think?
Yeah, so someone reached out to me a few months back and asked if they could use it for mice. And there's a good collection of mouse genomes available, sort of mouse pangenomes. So I constructed an index for pan mouse, which is called pan mouse one, which you can download in the same way, I believe, using the command line. But I would say, and this is maybe an advantage over some other methods, is that it's really not too difficult to build these indexes yourself. You will need a machine with a bit more memory. You might need for a pan-human and for building human indexes, you need closer to 20 gigabytes of RAM, which is not every laptop by any means. So yes, you can make essentially an index for any host, but I'm also very happy to you know to receive questions and help with that if you want to email me or otherwise contact me.
Thank you. Um, so the next one is, are there any specific things to be aware of when using Deacon, especially from the virus sequencing data?
From
Are you
virus.
going to demonstrate that?
Yeah, so I was going to just do a quick demonstration, but for viral sequencing data, so I would say that the panhuman index has been masked against viral minimizers. So from the RefSeq catalog. Um, so if I would say that if you are studying, yeah, if you were particularly interested in virus and phage, and you want to really maximize the retention of those and actually particularly viruses do often have overlap with the human genome. Well, or with human genomes as deposited in databases. Yeah, you could do some work, and again, this is you can do this using the Deacon command line, and there are some instructions in the README of how to do it. But basically, by default, it should work better than any other tool that I'm aware of. But if you really wanted to maximize its performance, you could perhaps mask the existing database with, you know, a custom collection of phage genomes that's more expansive than the one I've already masked it with. So yeah, you could certainly do that and look into the masking. It's Deacon index diff is the subcommand that you can use to do this, and I'm very happy to help anyone who's trying to make custom indexes.
And Bede will share his email address in the end of this presentation.
Oh yeah, I'll
And
put it in the chat.
yeah, how do you score your Deacon tool, 1 to 10, with
Uh,
compared to other tools?
um, Compared to other tools, well, I'd say the most interesting for me personally, I I think I a lot of other tools, most of the other Kamer-based tools are Kraken based, and then the others are mostly alignment based. Um, I'd say there's only one other tool that has a similar philosophy and achieves high accuracy in complex metagenomes, and that is this tool called Cleanifier, which I mentioned,
Mhm.
and it makes some very different choices, and in how it's implemented, but the result is broadly similar in terms of accuracy. I think it's a little bit less versatile. It doesn't. It's a bit more fussy about the inputs it accepts, and I'm not sure it accepts standard input, for example. Um,
Mhm.
but in terms of the accuracy of its output, I think it's really, um, yeah, I'm really impressed by it. So yeah, I'm sort of not really answering your question, but I think there's one other tool that's built with a similar philosophy, and I think either of them, either Deacon or that, is a good choice.
Thank you.
But I would say it does depend on your application because other tools have been developed for low complexity metagenomes, right? Maybe
Mhm.
for TB and other tools, some of these other tools that I've shown here, you know, they would achieve higher accuracy than Deacon in that context. Um, but my argument is that if you do not know what you're sequencing, you should expect complexity. Um, and some of these, and you need to, you know, you don't want to throw away bacterial genomes. That's my and you should yeah.
I got the message.
Running out of
Thank you.
words.
Yeah, thank you for the answer. We have two more questions that I will let you to do the demonstration. Would it be possible to share a minimalistic index that is both efficient and compatible with high accuracy?
Yeah, so that kind of relates to I feel like it relates to my what I was talking a little bit about earlier. Yeah, I'm wary of giving people too much, giving people the dilemma of choosing between quite similar indexes. But yeah, certainly, um, I could make a reduced version of the pan-human index that's half the size and nearly as accurate. Um, I would encourage you, if that's what you're after, I would encourage you to sort of email me or open a GitHub issue in the repository, just kind of explaining what you want and what your use case is, and then we can kind of go from there.
Yeah. I skipped the Sam's question, which is, would you suggest using Deacon for targeted sequencing experiment versus direct mapping to your target organism?
Uh, Targeted. So that's when you want to retain one thing. Um, yeah, I guess if you use alignment, you will be more sensitive. So
Yeah.
so KMAs will never be as sensitive as an alignment-based approach, but in some situations that can be an advantage. So yeah, it depends. If you need maximum sensitivity and you don't mind getting things that aren't quite your target, then you should use alignment. If you want to be a little bit more selective at the cost of maybe missing things with one mutation in them, then but yeah, to be clear, Deacon searches for by default, they're 31 mers, k-mers of length 31 across your sequence, and not everyone has to match. So you choose the threshold. It has sort of a default of 2 and 1%, both of which have to be satisfied, but you can change those to make it more or less sensitive.
Thank you. Last question is, how does Deacon complement adaptive sampling tools for ONT metagenomics, Oxford Nanopore technology metagenomics?
How does it complement? I'm not sure, but I don't know that much about adaptive sampling. Some of my colleagues have studied it in some detail. Um, I guess I was interested. I did think about adaptive sampling because I always wonder the extent to which bioinformatics limits the performance of adaptive sampling. But I think my understanding is that getting a bit faster at recognizing what's coming out of the pore is not really the limiting factor. So as it's my understanding that yeah, the performance of the bioinformatics is not really the limiting factor for how good your adaptive sampling is. Um, I don't know if that's the question that's being asked, but yeah, I'm happy to discuss again
Okay.
if I've got it wrong.
Thanks for all the answers and questions. So I'm handing over to you, Bede, for the demonstration. We still have
Okay.
10 minutes left.
Okay, so yeah, I was just going to give a really simple run through. So Deacon can do lots of things. Perhaps I should make my terminal a bit bigger.
That
Um,
would be very nice.
yeah, because I
I'm a
bet
little bit old.
this is a big screen. Um, so I guess I probably should have gone through the install process myself, but I do already have Deacon installed. So first of all, I mean, just go to the GitHub. Um, just talk about the different ways to install. I think most users will probably be most comfortable installing it using Conda or Mamba. So that's one option. And then if you have Rust installed on your system, you can compile it natively, which may provide slightly better performance, but probably won't make any difference. And then you've also got a Docker option. So here I've installed it because I've developed it on this machine. I've installed it using the Rust way. But anyway, I have this command now that I've installed it. Um, and then what I've also done is I've run this command that I had up earlier, which is, I'm just going to see what's in here. Yeah, so I won't download it again here, but you can fetch the index in a single command using deacon index fetch and then the name of the index. But actually, if you don't provide the name, by default, it will get the human index, the latest version of the human index. And you can see it's downloading that. I'm going to cancel it because I've already got one. And so essentially to use Deacon, the main command is called Deacon filter. And so you write Deacon filter, and I'm going to show the help here with -h to show you all the options. And so essentially you go Deacon filter, you then provide any options, and then you provide the path to your index, and that's the thing that was just being downloaded that I cancelled. So I'm going to go Deacon index, and I've got it stored in data. So, and this is the file name of the index by default. And then you provide a path to your sequences. These could be any sequences. They could be FASTA, they could be FASTQ. The quality isn't used directly. That's something to know. And they could be assembly contigs, or they could be raw reads. So here, what I'll do is I've got some reads here. And actually, this is the simulated reads of viral RefSeq. So essentially simulated 10x coverage genomes for 17,900 viral genomes. So hopefully, we don't map. If we do host depletion, we don't lose too many of these reads because these are microbial. We want to keep them. So I've got a few in here. So these are this is a FASTQ file compressed with Z standard, which is supported natively as well as gzip. So what this does by default, the filter command by default searches for things. So if we run this command now, we will output only things that match the index. Whereas actually for depletion, we want to put it into depletion mode, which you can see up here. So I'm going to go Deacon filter D, and then our index path, and then a space, and then our FASTQ path, and then we're going to say out dot FASTQ. Okay, so I've pressed enter and it's running, and it runs by default with 8 threads. So it takes a few seconds to load the index, especially the first time, and now it's running. And I'm hoping I've got enough disk space. I'm actually worried that that's why it's slowing down because I've got very little disk space left. But this is now writing our output. And actually, yes, I think it's slowing down because I'm running out of disk. Didn't think of that.
It's fine. It's fine.
Yeah, there
Oh
we go.
yeah.
Okay, so we've got confirmation. So I'm going to delete that before my computer crashes. That would have been a great ending to the call. See if I can recover that. But anyway, so yeah, that was running. So when we started that, even though the input was compressed, I think it was running at sort of 600 megabases per second. And so on a machine with enough storage, that should
Do
take.
you have an output, example output for us to see? It's typical
Yeah.
FASTQ file, right? Like, I mean,
Yeah,
there's
yeah, yeah.
nothing to see.
So I'll just sorry. We'll get something that runs through to completion. Yeah. Really clearly didn't think this through, did I? So what I'll do is instead of writing the output, I'm gonna send the output to the null device. So essentially, it's being written nowhere. You can also specify the output files. So remember, this can also support paired reads, and the README shows you how to output paired reads. But what I'll do here is I'll output a summary, which explains what it's doing. Summary.json, we'll call it. Oh, whoops. So now we're going to do the same again, but it's not trying to write anything, so it shouldn't run out of disk space. So yeah, now we can see it's running nice and fast, and it should be a bit quicker.
Oh,
Okay, so
that's
okay,
better.
so we can see exactly what it's done here, and actually I find this so often we talk about host depletion in terms of reads removed, but actually when you have ONT reads, they vary in length, so it's quite useful to know the actual the bases removed. So we've got lots of information here. We've got the parameters that we ran with. We've got our thresholds, our absolute and relative thresholds, which are really important parameters. Um, and we have what proportion of our input records passed the filter and what proportion of bases passed the filter. So you can see that for for these viral sequences, 99.98% of them made it through. So, that, yeah, as we would hope, most of them make it through. And I think that's a reasonable example. Um,
That's a good example. We've seen the output. That's really good. Okay. Yeah, thanks, Pete. Thanks for this amazing. Okay. There's another question,
oh,
but I don't think we do I mean, we have 2 minutes left. So
I'm happy to quickly answer another question
yeah,
if you
yeah.
like.
Can you feed it a directory instead of a single file?
No, you can't feed it a directory, but it's funny you say that because I've recently been implementing that in a kind of fork of Deacon, which does something else. So yeah, it's a really good question. Well, is that for like,
So
is that for instance, a
it's
barcoded
coming from
directory?
Tracy. Then is she saying that, then we specify for the file name that to be kept with a new file extension.
Yeah, so you can specify the output file extension. There are a few different ways to specify the output file extension. You actually you have to specify it. Um, but no, it's to answer your question, it doesn't accept a directory. Um, what you can do is create a named pipe to read from many files in a directory. I recently added support for that. But no, by default, you can't just give it a directory of FASTQ files. But if there's a use case where that would really help you, I'm very happy to think about it. And please do send me an email or open a GitHub issue.
Thank you. So, due to our time constraints, thanks, Pete, for this amazing demonstration and the presentation as well, and answering all the questions. So, we forgot to include your email. Could you write down email to the chat if possible
Yeah,
for anybody who wants to ask a question, or they can go to your GitHub page and they can find yeah, your
yeah.
communication
You can
channels as
also
well.
find me on Blue Sky. And actually, if you go to the Phage Blue Sky, you'll see they posted something with my handle in it there. Yeah, feel free to email me, contact me on Blue Sky, or open a GitHub issue. And yeah, thank you so much, Gultigan, for allowing me to speak and share my opinions, and for managing these questions. And thank you so much, everyone, for all of these questions and for listening today.
Thank you. Amazing. It was an amazing webinar. So please keep in touch for the next upcoming webinars as well. And do not forget to sign up for Phage membership as well. So see you in the next one. Thanks, Pete.